Welcome back, students. This is a video lecture on Chapter 14, the focus of which is to compare forward genetic techniques with reverse genetic techniques. The chapter, being divided into 4 parts, helps students understand firstly forward genetic screening. Then it looks at how using recombinant DNA technology aids us in deciding how to elucidate the structure and function of genes. The third part looks at reverse genetics and investigates how those techniques have helped us to identify gene phenotypes. And the last section looks at transgenes and how they can be utilized in dissecting gene function. The word screening in genetics is equivalent to filtering. If you have 10,000 individuals and you want to filter out the ones that are not part of a study, then what you're doing is you're screening that population to only identify those that are of interest. With the advent of Mendelian genetics, the only process available to scientists in the 20th century was forward genetic screening. Here, individuals would be identified as mutant initially, and then they would be investigated to figure out which genes were defective inside the mutant. The goal being to understand what the wild type allele of that gene does and how it interacts with the biochemistry and cell biology of that particular organism. Thus, the starting point was the existence of a mutant. The full power of this approach was revealed when scientists such as Quinn and Benzer first suggested that genes were involved in behavioral traits. People didn't believe it at the time, or thought it was too complex to investigate. However, in their simple experiment on the fruit fly Drosophila, they were able to show that certain mutants existed in the population that were deficient in learning and memory. What they basically did in the experiment was to expose flies to an odor and thereafter subject the cage to an electric shock. And those flies that learnt that the presence of the odor would entail an electric shock thereafter actually flew off into the air so as to avoid the electric shock. And they were able to identify those flies that were not able to associate the two factors together. These were the mutant flies, and from those experiments they identified 2 genes. One they called dunce and the other one they called rutabaga. Subsequent biochemical characterization of both these gene products revealed that they are involved in the production and or degradation of cyclic AMP. Cyclic AMP is a very small and important molecule inside cells. It acts as a messenger. It conveys information from one system within the cell to another, therefore leading to some outcomes. Thus, one can imagine in neuron cells of these flies' brains, they are unable to transfer information from one area to another, thus mitigating the effects of learning. Subsequently, further research into the biochemistry of learning has revealed that cyclic AMP does indeed play a very important role with respect to learning memories. In particular, an associated protein known as cyclic AMP response element binding protein, CREB for short, turned out to be a transcription factor that was known to turn on certain genes. The important thing about forward genetic screening experiments is that they are unbiased, and they have no prior knowledge of the molecular function of any particular gene or gene product. Therefore, the mutant that you have is the mutant that you have. Unfortunately, the presence of natural mutations may not be great enough to conduct a full investigation. Therefore, scientists began to explore new ways of increasing the rate of mutation safely in organisms. And that is the process of mutagenesis. We'll look at the methods of applying a mutagen in a few slides, but for now, the important thing is that once you have lots of mutations in a population, you have to then screen that population for whatever you're looking for, the phenotypic defects that are part of your study. And only a small fraction of those individuals will have the mutation of interest, a tiny fraction. Mutagens are substances or techniques that increase the rate of mutations randomly throughout the genome of the organism in question. The conditions are adjusted with the goal of introducing mutations on average into every gene one achieves that goal, we call that saturation mutagenesis. More so in the past than today, selecting the right organism was very important. Certain organisms, known as model genetic organisms, have been used because they offer certain advantages over their counterparts. Model organisms should possess certain characteristics which are conducive to research. Some of the more paramount concerns are: can that organism be reared in the laboratory? Does it have a short generation time? Will it produce lots of progeny? Is it easy to cross in matings? There are a number of other criteria, but these are the main ones. If the organism chosen is diploid, then one has to rear this organism to a point that it is homozygous at all loci, i.e. pure breeding, before conducting the experiments. And of course, it's always advantageous to use the simplest organism possible if one has a list to choose from. Since DNA is the fodder for all mutations, and the definition of a mutation is a change in the DNA, we learnt in the past that DNA can be changed in many different ways. You can have simple nucleotide substitutions, insertions, deletions, inversions, duplications, etc., etc. So the choice of mutagen utilized depends on the research organism that you have chosen and the type of mutant alleles that you desire. Of the various choices of mutagen available, chemical mutagens seem to be the most suitable for introducing mutations at a frequency suitable for investigation. So applying a chemical mutagen only takes a few thousand individuals in the population to have at least some of them carry the mutation in the gene of interest. However, cloning genes mutated by chemicals can be tedious and laborious. A useful alternative to chemical mutagens is the use of transposons. As we may have learned in the past, transposons are autonomously replicating selfish pieces of DNA that can move around genomes. Unfortunately, transposons are used when chemical mutagens don't give the appropriate desired results. As we'll see later in the chapter, transposons can be used to induce mutations, but because of their locations, they will generally result in fewer mutations. So saturation is difficult. Saturation of mutations in the genome is difficult. However, one advantage of transposons is that they themselves are DNA, and that DNA can be uniquely identified and tagged so that it makes finding and cloning the neighboring genes relatively easy. Regardless of the method of mutagenesis employed, in most cases, one has to go further and perform genetic crosses in order to isolate individuals with homozygous or heterozygous configurations for the gene or genes of interest. Table 14.1 lists 3 common mechanisms of mutagenesis and how they interact with DNA. Students are expected to know these 3. So chemical mutagens, an example is EMS, ethyl methane sulfonate, that normally causes transitions. And as you know, transitions are where the base stays within the same family of chemical classification. So a purine to a purine or a pyrimidine to a pyrimidine. And they introduce stop codons frequently. And they can also destroy splice sites, normally resulting in a loss of function mutation that can be null, hypermorphic, or hypermorphic. A different type of mutagen is radiation, especially X-rays and gamma rays, and they normally cause rearrangements of DNA. Deletions, inversions, and translocations are most common. And they normally result in a loss of function also, but this time mostly because of null mutations. But occasionally there can be a gain of function mutation. And finally, we have these biological agents known as transposons, and they can insert themselves, and they do so at a pretty low frequency compared to chemical mutagens, and they also normally result in a loss of function of the null type mutation. Once these mutations are introduced into the population, in order to discover and purify these mutations by filtering, one has to conduct a number of genetic manipulations, breeding experiments basically. And you're familiar with these because this is exactly what Mendel did in his first experiments. There are different variations on what must be done depending on the nature of the mutation. So let's start at step number 1. So this is a strategy for identifying dominant and recessive mutations. So these newly introduced dominant mutations can be identified in the F1 generation simply by breeding the mutagenized males with wild-type females. The offspring in the F1 will immediately show the phenotype that we are generating. And from those we can select the individuals that are of interest to our project. One thing to note, dominant mutations are relatively rare because they will require the biochemistry to work in a way that experiences a gain of function. So these are gain of function mutations. Figure 14.2a is very important to understand because it's the foundation of subsequent figures. In this particular example, we're looking at a screen to identify dominant mutations. The process begins by taking wild-type males and subjecting them to mutation so that the genes are randomly mutated. In this example, this box here indicates that the middle gene carries a mutation in the heterozygous condition, and those males will then be able to form gametes, and the gametes will segregate into those that carry the mutation and those that don't. And they will be bred, these males will be bred with a normal female from the population, and her gametes will be all positive, wild type for all 3 locations. In these circumstances, you should be able to predict that half the offspring in the F1 will be carrying the dominant mutation and the other half will not. Because the phenotype of half the population is obvious, it's now a simple matter of picking those individuals that have the phenotype that you want to investigate in your research. The situation when trying to obtain recessive mutations is a little bit more complex, but guess what? You should be familiar again from Mendelian genetics on how this would pan out. Please do not be intimidated by figures as complex as they look, because they're really not that complex. Let's just concentrate on panel B. Panel B is different to panel C because this looks at individuals that cannot self-fertilize, so humans and flies. And then over here we'll look at individuals that can self-fertilize, plants and some types of invertebrates. The beginning part of the strategy is the same as in the previous example. We take males, we mutagenize them, and then we breed them to wild-type females. Only the F1 males are then used in the next set of experiments where they are bred to normal wild-type females. The resulting F2 generation is interbred amongst themselves, and then the F3 generation is examined for the presence of homologous mutations. And these are the ones that we can then filter from the rest of the population. So a multi-generation task in order to obtain homozygous recessive mutations. The number of steps required in individuals capable of self-fertilization is fewer because by the F2 rather than the F3 you can obtain these homozygous recessive mutations. Basically the steps are similar at the beginning. You take males or one sex and then you mutagenize them and you breed them by themselves because they're capable, and then you can immediately see that 1/4 of the progeny will be homozygous recessive. One can then screen this population for the mutations of interest. Moving on to another system of selecting for mutants, the use of balancer chromosomes. I'll let the video that's coming up next speak for that, then we'll come back to this.
Balancer chromosomes are fake. This is something that I missed when I first learned this, and I think it's really important for you to understand that these are engineered chromosomes. The wild-type chromosome has genes that are in order. They're always in this order so that when the chromosomes align, crossing over can take place. And this increases genetic diversity. You want this in a population because statistically it will increase survival. A balancer chromosome, you can see here, has the same genes. It's just that some of them are backwards and out of order. So all genes are there, but they're just not in the right place and they're not in the right order. So when these chromosomes try to line up and cross over, it's not possible because they won't actually have homologous regions on the chromosomes. So crossing over does not occur. And this is a real strength to maintaining a mutant fly line. You don't want genetic diversity. We want the flies to be heavily inbred and we want them to be genetically very similar. We do not want to increase the genetic diversity of our population.
Thus, the use of these artificial balancer chromosomes allows genetic manipulation without too much fear of crossing over. Here are three important features of balancer chromosomes. They have one or more regions of inverted repeats, and that's to prevent the crossing over mechanism from forming synapses. A recessive allele resulting in lethality. So if any individual gets 2 copies of the balancer chromosome, it won't survive. And they also contain a dominant mutation producing a visible phenotype, so that segregation of these chromosomes can be seen as we follow them through different generations. Why does one want to use balancer chromosomes? Well, the answer is given here. In the previous set of experiments, namely this one, it's difficult to produce enough mutations through all these different crosses to satisfy the needs of the scientist. So if the species is amenable, then balancer chromosomes are preferred, and they circumvent the lack of mutations that are seen in the F3 screen. Here is an example protocol utilizing balancer chromosomes. So flies are first treated with EMS to induce mutations, and then they are mated to female flies carrying the balancer chromosome. So that's important. They're not wild type any longer, they carry the balancer chromosome. Since these females are carriers, then they only have 1 copy of the balancer chromosome. The homologous chromosome is normal, completely normal. The goal of the experiment is to transfer to the F1 males the mutagenized chromosomes from their fathers and the balancer chromosome from their mothers. And that's what we're selecting for in the F1. So once we've selected for these F1 males, they are then mated with balancer females again. And this time we'll score the males and females in the F2 generation. Let's pick up the figure right here. These are the F1 males, and they're bred with females of the same type as their parents. And they then produce this genetic cross with the female gametes on top and the male gametes on the side. You will notice that this box is empty because these females that have 2 copies of the balancer chromosome, they don't survive. The F2 males and females are interbred to produce the F3 generation. We can then select heterozygous individuals or homozygous mutant individuals. We still have to address what happens with haploid individuals. Populations of haploid individuals are advantageous because the gene is present in a single copy, therefore they're haploid. And that gives rise to easily identifiable phenotypes. There are some disadvantages of haploid organisms in that they can produce lethal alleles that will kill the individual. To circumvent this, one thing that geneticists have realized is that by using conditional lethal mutations, some phenotypes are able to grow in one set of conditions, but if you move the organism to a different set of conditions, then the individuals will die because of some protein interactions with the environment. These situations can be quite complex, but if we look at it from a simple perspective, we can say that the product of that gene may not be needed or necessary in the first set of conditions, but when you move the organism to the second set of conditions, it is required. So the first set of conditions, the ones that allow the mutated organism to grow, are called the permissive conditions. But when you relocate that to another set of conditions, those are the ones that cause the phenotype to be affected by the mutation and thus die. Those are called the restrictive conditions. Depending on the nature of the mutation and its biochemical outcomes, Some conditional lethal mutations, once identified, can be rescued by supplying the ingredient that's missing in a biochemical pathway. In other cases, in different circumstances and different scenarios, the conditions may be something to do with temperature. At one temperature, the mutant doesn't reveal itself, and at the different temperature, the mutant reveals itself by killing the organism. These temperature conditional mutations were key in allowing us to discover the regulation of the cell cycle in eukaryotes. In the early experiments, yeasts were used to replicate what happens in other creatures, and the mutations that they studied, they fell into discrete categories or groups, depending on the stage at which the cell cycle was arrested at the restrictive temperature, normally the higher temperature. And from these we came up with the understanding of checkpoints in the cell cycle that we've covered in previous lectures. So experimentally, the yeast were mutagenized and then plated onto plates where they could grow at low temperature. And then copies of these colonies were transferred onto second plates, which were then incubated at higher temperatures, 36 degrees. And any colony that did not grow on both plates indicated that that colony came from cells that were mutant. And they were mutant for these temperature-sensitive mutations. To investigate the missing colonies, you have to go back to the original plate and then pick those colonies and then do a biochemical analysis or genetic analysis on those. And those types of analysis have revealed that yeast cells have certain phenotypes that they stop at, that they arrest at, if they are subject to different temperatures. And those correspond quite nicely with the cell cycle that we have learned in the past. So the arrested points indicate that there are three major checkpoints which were associated with these gene mutations. And further research has shown us the association between these mutations and the cell biology. For instance, the CDC16 mutation in yeast has been correlated with this phenotype, which now suggests that there's something in that mutation that interferes with DNA synthesis. So that cell will stop right here at this checkpoint between S and G1. Why do scientists wish to perform forward genetic experiments? That's a big question. There are many reasons for doing this, but some of those would be to identify dominant versus recessive mutations. Others would be to identify how many different genes are involved in a biochemical process. And then finally, how many different mutant alleles can exist for each gene, and how are they correlated with the phenotype? How is one able to determine if an allele is dominant or recessive, sex-linked or otherwise? Well, these things are assessed during the mutagenesis experiments and confirmed in the same way that Mendel confirmed things by breeding different combinations of individuals, whether they're haploid, sexually reproducing, or asexually reproducing. Simply by looking at the ratios within the offspring, one can have a great understanding of the relationship between the biochemistry and the genetics in terms of dominance and recessiveness. With respect to the second question, how many different genes are controlling that particular character, we can do complementation testing. In complementation testing, if two individuals that carry the mutations are interbred and their offspring are wild type, then we can say complementation has taken place and that the two mutant alleles must be in different genes. If, however, the breeding experiment results in mutant offspring, then the mutations must be affecting the same gene, and thus there was no complementation. The answer to the third question is straightforward. It relies again on complementation analysis. For every time a complementation test fails, this type, and we find different phenotypes, we can then understand that each one of those phenotypes may be a different mutation within the same gene, i.e. these are mutant alleles. Incidentally, the recovery of multiple alleles is an indication that saturation has or has not been achieved through mutagenesis. The next section deals with modifier screens. What are we trying to answer? Well, we want to know if two genes are interacting with each other and therefore interfering in complex ways. In essence, we're looking at epistasis, or forms of epistasis, because a genetic modifier screen is simply used to see if mutations in the second gene will modify the phenotype of the effects of the first mutation in the first gene. There are two main possible types of outcome, a positive outcome and a negative outcome. In a positive outcome, the two genes are enhancing each other's effects. So they enhance the mutant phenotype. And in the second type, we have a suppressor screen, where the second mutation suppresses the first. An extreme example of this very broad range of interactions between genes is that of synthetic lethality. In this case, two genes interact with each other to cause a set of alleles to produce a lethal outcome. Each mutant allele in itself doesn't cause death, but the combination of two of them will. Therefore, synthetic lethality. The example given here is in Drosophila, the fruit fly, where the combination of a mutant form of the prune gene as well as the prune killer gene results in a lethal outcome for those individuals. Figure 14.5a uses classic genetic symbology in order to illustrate this example. First thing to realize is that the symbol for male and female are used. So females have two chromosomes, and on one chromosome, this location, which will be equivalent to the prune killer location, is wild type. So she's homozygous wild type for prune killer. On the other chromosome, and you can tell that's a separate chromosome because these two bars are separated from each other. So on a different chromosome, a second gene is homozygous mutant, and this is the gene for prune, the prune phenotype. She's mated with a male. Now the prune phenotype, we can tell now, is on the sex chromosome. So the sex chromosome of these flies in the male is hemizygous, therefore there's only one copy, and because of that one copy being mutant, this fly will have a mutant phenotype for prune. And at the second locus, on the second chromosome, this is telling us that this is not a sex chromosome, this is an autosome. So here, he has a double mutation. When these two flies are bred together, the gametes produced here will be PN plus, and there'll be two types of gametes produced here, PN plus and killer prune, and the Y chromosome and killer prune. So all the females that are produced will be wild type. Or would they? Well, this tells us, when you look at the phenotype of these females, they are wild type for prune, but they are killer prune for the second locus. So this capital K indicates that this allele is dominant to the wild type. So this is a very important point. The capital K indicates it's dominant. The lowercase p indicates that this allele is recessive to the wild type. And in all the males are indicated by having a Y chromosome, they would be prune and then killer prune. Because you have a double mutation, this will be classified as synthetic lethality. So all these males will die. Can we use biochemistry and cell biology to help us understand this mechanism? Indeed we can. So the way that synthetic lethality works, it uses two main mechanisms. In one mechanism, we have two pathways that each feed into an essential biological function. The two genes act in parallel, and complementary pathways are needed for some essential function. As long as one pathway is fine, you will get some type of phenotype that's survivable. So a defect in one pathway will still allow the individual to survive because pathway B is still active. And the converse, a defect in pathway B will still allow the individual to survive because pathway A supplies the essential function. So that's an example of parallel. We call that genetic redundancy, by the way. I didn't mean to slip it in quickly, but that's an important concept that we've seen in other chapters that we hadn't given a name to. So we're giving that system a name right now. So genetic redundancy, when two pathways lead to the same biological function. The second pathway, or in the second scenario, both genes act along the same pathway, with one acting maybe at the beginning of the pathway and the second acting at the end of the pathway. So a deficit in the beginning of a pathway, for instance here, will reduce the amount of C2 being made, but there's enough C2 being converted to something else that the individual survives. Or you could have a deficit in the second half of the pathway. So the first part of the pathway is fine, and the second part is reduced. And the word reduced is very important. And if the second pathway is reduced, you have the same outcome as the first pathway. The amount of final product may be 50% of what it has to be, but enough to keep the organism alive. And in the final example, we have disruptions in both pathways. And the organism cannot survive that because this reduces it by 50% and then this mutation reduces it by another 100%, so there's nothing produced. There's not enough chemical, and that's not going to allow the individual to survive. Applying the cell biology angle to this conundrum, we can do that. So genetic redundancy, this one here, we can explain by saying that the two genes encode proteins with very similar functions, so that one can substitute for the other to some degree. They can't fully compensate for each other, so that the mutation is either 50% or some percentage of the original. And the outcome is a mild phenotype. Now that all depends on how similar they are. If they're very similar, then the mild phenotype may be very mild. However, there may be alternative biochemical and cell biology interactions that have nothing to do with one function being replaced by another, and these can be of many, many different types, where possibly one protein interacts with another in a complex manner. But regardless, they can still be found through modifier screens of the nature that we've been talking about. So let's conclude this section of the chapter by summarizing what we've learned. We learned that you can increase the rate of mutagenesis by artificial means, therefore producing more mutants than naturally occurring. We also learned that by using the right type of technology or the right type of mutagen, you can impact the DNA in the appropriate manner to generate mutations. We also learned that screening, i.e. looking and seeking the type of mutation that you need, can be assisted by genetic crosses and understanding how balanced the chromosomes work. Sometimes you may have to resort to transposable elements in order to achieve your end result. Regardless, the process we've been looking at is forward genetics, where you start with a mutation and then you try to figure out what's causing that mutation. Hopefully it's one or a combination of genes. Let's move on to using technology to help us speed up or generate new outcomes that are not possible through natural systems. It's important to understand that recombinant DNA technology, used to be called genetic engineering, is a tool, nothing more. And this tool allows geneticists to take a giant leap forward in trying to elucidate the nature of a particular allele of a gene or set of genes. The main leap is from the mutant phenotype to the sequence of the DNA of that particular segment of the chromosome. The term transgenic organism, which elicits in lots of people's minds the images of monsters, is basically the term applied by geneticists to when somebody takes two DNA sequences, either natural or artificially made, and puts them together and then introduces them into an organism. That organism would then be referred to as transgenic. These transgenic molecules and transgenic animals can be very powerful in genetic analysis. Let's talk about DNA libraries. A real brick and mortar library is simply a curated place where knowledge is kept. The better the library, the more knowledge it encompasses. A genomic library is similar. The brick and mortar would be equivalent to the host cells, and the books and the knowledge would be equivalent to the fragments of DNA that come from an external source. Let's give you the same knowledge in a different way. If I took all my DNA and I fractioned it up into small pieces by using sound waves, my DNA will be broken into billions and billions of small fragments. If those fragments were then inserted into a bacterial chromosome randomly, and all the bacteria that contain my fragmented genome were then grown in a single place, that whole collection of bacteria would be Dr. Dulai's genomic library. So within those fragments, you should be able to find every aspect of my chromosomes in some clone or another. And what is a clone? It's a colony of bacteria growing in that environment. What would that be? Well, in the case of humans, we can clone our genes into bacterial or yeast vectors, and the vector is the name of just a carrying molecule. So the yeast chromosome will be a vector for my DNA. And if that yeast chromosome was reinserted back into yeast cells, then the full collection of my DNA in all the yeast cells will be the genomic library of Dr. Dulai. And in the future, you could go and pick a clone that contains a particular sequence of mine and then grow that up in a flask and then perform some experiments in characterizing my genes. So genomic libraries represent the entire genome of an organism. If, on the other hand, you went to my liver cells and you wanted to just know which messenger RNAs were being made at that moment in time, then you could deconstruct my liver cells, isolate the messenger RNA, and then from the messenger RNA make double-stranded DNA using a sophisticated set of enzymatic techniques. And once you have copies of my messenger RNA as double-stranded DNA, that can then be inserted into the vector to make another library. But in this case, because you're using messenger RNA, you're not using genomic DNA, you have something called a complementary cDNA library. So that represents just a subset of my genes that happen to be active in that cell type at that moment in time. And they can be useful when studying biochemical processes. So genomic libraries are the complete genome, and complementary cDNA libraries are just representing the messenger RNA active at that particular time. Next, we're going to discuss two approaches to identifying the physical nature of specific genes. The first method is by complementation. In this regard, the mutant phenotype is assumed to be recessive, and the presence of a second piece of DNA coming into that cell will enable the gene function to be restored, therefore negating the effects of the mutation. This is best illustrated by using yeast cells. Each of these yeast cells has a mutation that prevents it from growing along the cell cycle. So all these cells have the same mutation. The next step in the analysis is to subject all these cells to some type of transformation where they pick up these plasmids containing a copy of a gene that expresses itself. So now each cell has the potential to pick up different fragments of the genome of this yeast from a normal condition. So in this case, it's picked up a fragment of a normal gene, but that gene has nothing to do with the mutation that the cell has. So the two will not complement each other. And nor will this, nor will that, because these fragments are coming from different chromosomes that have no correspondence to this particular mutation. But in this cell here, just by chance, the fragments in the library are able to complement the mutation on the chromosome of this cell, thus restoring the biochemistry to the wild type. How are we able to screen for that? Well, we plate all these cells at the permissive temperature, and then we move them off to the restrictive temperature. And at the restrictive temperature, none of these other cells can grow because there was no complementation. the original mutation was not overcome, and these cells cannot grow at the higher temperature. But in this one colony produced from this cell, it is seen growing on the restrictive plate. Therefore, we know that that colony contains a complementation between the original mutation and the incoming fragment. What we have to do now is characterize the incoming fragment, and then we'll have the identification of the corresponding gene. One would assume that these days it's very easy to sequence DNA. It is, but sometimes there are complex relationships going on inside the cell that the sequencing process may not reveal. One has to discriminate between wild-type polymorphisms, these are natural polymorphisms of a gene that exist in the population, and the link between the mutation and the phenotype. So these are causative mutations versus wild-type polymorphisms. How do you know which one is which when you are sequencing a gene? That's not always straightforward. And also, the process of mutagenesis can produce hundreds of new mutations, especially sometimes inside the same gene. The trick in this case is to take the mutant strain, the mutant cell, and backcross it with the wild-type strain, or the wild-type cells. And that can separate the one causative mutation from other mutations. This principle is nicely illustrated by figure 14.7. The mutant individuals are crossed with the wild type, and then the F1 are interbred, and the resulting F2, the mutant phenotype, is then sequenced. All these individuals are sequenced, and that's what these arrows indicate, maybe hundreds if not thousands of them. And then the computer will line up all the sequences that it generates from each individual, and then we can identify any commonality that exists between all these individuals. And any other mutations that happen to be present, as well as polymorphisms, will be diluted away because there won't be a consensus. So the causative mutation will be present in all the sequences, in this case this one. A plot of data from different regions will generate outcomes that will identify a hotspot, and that hotspot should correspond to the location of the mutant that's common in all these individuals. That concludes the second part of the chapter. So what can we summarize? We can use technology to take us from mutations to gene sequences. The mutant individuals are crossed with the wild type, and then the F1 are interbred, and the resulting F2, the mutant phenotype, is then sequenced. All these individuals are sequenced, and that's what these arrows indicate, maybe hundreds if not thousands of them. And then the computer will line up all the sequences that it generates from each individual, and then we can identify any commonality that exists between all these individuals. And any other mutations that happen to be present, as well as polymorphisms, will be diluted away because there won't be a consensus. So the causative mutation will be present in all the sequences, in this case this one. Let's move on to part 3. Part 3 is basically reverse genetics. So in forward genetics, we start with a mutation and we want to find out what gene is responsible for that mutation and its alleles. In this case, we start with DNA sequence, and then we want to discover what genes that DNA sequence is part of and what do those genes do in terms of phenotype. So we're going backwards with respect to the historical investigation of genetic phenomena. This approach has come into its own because of the huge amounts of data now that exist in databases around the world based on sequencing of genomes of many, many creatures. There are many options to discuss, so the textbook reserves itself down to 4 technologies that are pretty powerful. We begin with the most powerful at this time, the use of gene editing. Just like you have a word processor that can edit content in terms of a file, we can have now technology that can edit DNA in this context of DNA sequence. The process relies on several features of cells. The first is that cells have the ability to cut DNA internally inside the molecule, and those enzymes are called DNA endonucleases. and they target their location by matching a small part of the surface to that of the DNA base composition. And we saw this earlier in the example of restriction enzymes. So restriction enzymes are enzymes, proteins, that are present in bacterial cells that can cut DNA internally by recognizing up to 4 bases, as few as 4 bases. Once the DNA is cut, it's handed off to a second system, and that second system could be one of two. It could be non-homologous end joining, or it could be homologous recombination. And that depends on the phase of the cell cycle that the cell that's perceiving this treatment is in. If non-homologous end joining is left to do the second stage, then that can introduce small deletions at the site of the break introduced by the DNA endonuclease. And because those deletions can impact the reading frame, we may end up with loss of function mutations. But occasionally we get gain of function mutations. With the homologous recombination system, the system can exchange DNA fragments by taking externally supplied DNA and recombining that and replace the internal sequence, therefore swapping out genes is what we're saying. There are two approaches to achieve this site-directed mutagenesis. The first uses an old system, where a protein nuclease can be tethered to a DNA-specific sequence, and that then has to recognize a binding domain on the DNA that it then cuts. However, this is now being overtaken massively by the second mechanism, that of using a nuclease that uses an RNA molecule to target a site on the DNA. And this is all the rage at the moment. This year alone, up to 50,000 papers have been published that are concentrated on using this particular gene editing technique. The entire process, the entire technology is based on the CRISPR-Cas9 system. What is CRISPR-Cas9? Well, CRISPR-Cas9 is a region of a chromosome and the activity of a nuclease combined together. Historically, in the 1990s, off the Mediterranean coast, scientists found these archaeal microbes, archaea basically, that show repetitive DNA along their chromosome. And that DNA could change depending on the conditions in which the bacteria found themselves. The arrangement of the DNA in this location consisted of repeats separated by spaces. And in some cases there were many, in other cases there were fewer. And this region of the chromosome was termed CRISPR, and that stands for clustered, regularly interspersed, short palindromic repeats. So you break it down from the back. Palindromic means a word spelled the same forwards and backwards. So a piece of DNA that could spell the same forwards and backwards is called a palindrome. And these are short palindromic sequences that are repeated multiple times. But interspersed between them are these clustered repeats. So the repeats are interspersed between the palindromic sequences. In addition to this region of the chromosome, just upstream there are additional genes that only occur once. And those are called the CRISPR-associated genes, or the Cas genes. And they include the information for producing the endonuclease, the enzyme, the protein. It is now known that in these bacterial cells, the natural function of the CRISPR-Cas system is to defend the bacterial cell against invading nucleic acids, namely from viruses. The spacer sequences are basically historical records of old infections that this particular cell has beaten. So when a virus attacks a cell, sometimes the virus wins and the cell dies, and that's the end of the system. But if the bacterial cell wins the battle against the virus a small section of the virus's genome is kept and then inserted into the spacer region on the CRISPR locus. So that becomes a memory of the viruses that had come across the path of this bacteria in the past. But not only that, when this bacteria replicates and divides into daughter cells, it copies this region of the chromosome, therefore passing this as a memory to its offspring. And if you think carefully, that last part will be equivalent to our immune system using T cells and B cells, where we have a memory of past infections. Figure 14.8a shows us the arrangement of the CRISPR locus along the chromosome of bacteria. So we have, in this case, the cassette that makes the tracer RNA. That tracer RNA has a structural role in holding things together. Then we have the CRISPR locus itself, and the repeats are indicated in different colors in this case. And when that region is transcribed, it produces a precursor CRISPR RNA, which is then cut by endogenous RNAs into the right length. And these are then combined with the tracer RNA and loaded onto the protein, the Cas9 protein, which is made from one of these genes here. The whole system comes together, and the CRISPR RNA from this region here has a common area that sticks to the tracer RNA, base pairs, and then it has a unique single-stranded region that's color-coded here that sits across the Cas9 protein. Then the Cas9 protein is ready to go hunting. So one place that talks about this system says that the protein is the hardware which allows this system to track down double-stranded DNA and then scan it for equivalence to its sequence. And the RNA, the CRISPR RNA, is the software that allows this piece of hardware to be programmed with what it needs to go and find and destroy. So in a cell, you may have 50 different types of Cas9 protein, and each one may be loaded with a variation of one of these CRISPR fragments. So thus, simultaneously, you could be scanning the cell, looking for complementary sequences to the RNA, and any DNA, whether it's viral or otherwise, that identifies a match will be cut by the endonuclease activity of the Cas9 protein. Figure C simply shows us that once foreign DNA is recognized, a small fragment of it is cut and then integrated back into a new spacer that increases the number of repeats inside the CRISPR locus. This would therefore be the memory component of the system. What Jennifer Doudna and her colleagues were able to do was to take the tracer RNA and the CRISPR RNA and combine them into a custom molecule, which they called the guide RNA. The front part of the guide RNA is customized to match the target sequence that the researcher wishes to change. The back end of the guide RNA contains common elements that are needed to bind to the Cas9 protein. Once both elements are introduced into a cell, either by genes or by direct injection, the cell then has the ability to form the Cas9 system. The Cas9 system comes together with the guide RNA sitting inside the Cas9 protein. This completed ribonucleoprotein then can travel throughout the cell, making contact with double-stranded DNA, trying to find a region that's homologous in terms of sequence matching, nucleotide matching with the RNA. Once that region has been found with some degree of compatibility, the region of DNA is cut on both strands. Thereafter, natural repair mechanisms in the cell take over. In the case of double-strand cuts, the non-homologous end joining mechanism is normally available full-time, to introduce small deletions into the ends of the DNA before splicing it back together again. That normally inactivates the gene that was part of the cut region. Alternatively, if you supply your own piece of DNA as the third item, along with the guide RNA and the Cas9 protein, then doing another mechanism that the cell has called homologous recombination, the external DNA can be spliced into the gap created by the double-strand cut. And that introduces a new copy of a gene into that location. The good thing is that you could have multiple guide RNAs simultaneously present alongside multiple Cas9 proteins. And simultaneously, the DNA will be cut at each one of those locations, therefore introducing or deactivating genes en masse. Therefore, combining all these techniques together, you come to this conclusion that the CRISPR-Cas9 system may be applied to gene therapy or to agriculture, often with some ethical considerations to examine. In the literature lately, someone in China has used this system to alter the DNA of zygotes, baby girls, and their DNA has now been changed to an alternative form that prevents them from capturing any HIV infections. If extraneous DNA from outside the nucleus or cell, is incorporated into a gene itself, that normally leads to the reading frame of the gene being disrupted. Or if it occurs in the regulatory component, it could cause the regulation to be disrupted. In most cases, the incorporation leads to a loss of function allele being generated. This method is pretty inefficient, especially for complex eukaryotes like you and I. Therefore, alternative resources have to be utilized by scientists. Luckily for us, we can access something known as a knockout library. A knockout library is a collection of clones generated with mutations in all the genes so that all those alleles are non-functional. And instead of making your own, you can buy these from resources such as biotech companies and get them from other scientists, such as for yeast cells and for bacteria, where every strain has a single mutation in a different gene. Table 14.2 lists some of the more commonly available sources and the mechanisms used to generate those knockout libraries. Returning our attention to recombination, there's two types of recombination that can be witnessed when it comes to generating resources for genetic investigation. One is called illegitimate recombination, and we'll talk about that next. And the second is homologous recombination, which we have some knowledge of from prior lectures, but we'll mention it again in this context. With illegitimate recombination, the external piece of DNA integrates into a random non-homologous location within the chromosome of plants and animals. With homologous recombination, the piece of exogenous DNA, external DNA, is incorporated by recombining into a homologous site by matching up DNA, and that occurs more commonly in bacteria and fungi. Further, it depends on the structure of the two pieces of DNA as to what happens. If the exogenous DNA is linear, or if the exogenous DNA coming in is circular. In addition, it depends also on whether you have one single crossover event or multiple. Let's look at the example of a circular chromosome being integrated into an organism's internal chromosome. So here's a yeast chromosome, and here we have a plasmid. A plasmid is the external introduced piece of DNA, and it contains a selectable marker inside the gene. So this is a mechanism to determine if this fragment of DNA has been picked up by the yeast chromosome. So if no crossover takes place, normally what happens is the plasmid will be lost from the cell after a period of time. Crossing over requires homology between the target region and the region on the plasmid. Once that homology has been identified, enzymes cause a crossover event where you can see, if you follow the cursor, the DNA that's created follows this pattern here. So the entire plasmid can become incorporated into the target gene. And the target gene is disrupted. You can see the light green on either side. And the plasmid is then inserted into the chromosome in an orientation. That's one possible mechanism. With a double crossover, you can see that the majority of the plasmid is not incorporated into the chromosome, and also the target gene has lost fragments that are picked up by the plasmid. So there's a reciprocal exchange of genetic information. So this time, if you follow the sequence, you go along like this. You jump up here into the plasmid and then back into the chromosome. So now you have a part of the gene on either side and then the target selectable marker in between. The same sequence of options exists with linear exogenous DNA. So in this case, we have homologous recombination that leads to the production of this. So you follow the cursor again, you jump up here, and you go along this way. And assuming that the centromere is on this side. So that is the newly made chromosome. And this part will be lost, and so will this part of the plasmid, because it doesn't contain any genetic information to retain that molecule inside the cell. In a double crossover, only the central portion of the plasmid, the linearized plasmid or the exogenous DNA, is incorporated into the target region on the chromosome, but both chromosome arms are left intact, attached to the original piece of DNA. Therefore, scientists have options and choices to make in order to determine which of these events is required and which one actually took place. Moving on to something known as gene knockouts. A knockout is basically a maneuver in which the insertion of information into a gene deletes the entire coding region of that gene. Therefore, it knocks it out, makes it inactivated. And a knock-in will be the opposite, where you insert something that increases the functionality of that cell by introducing new exogenous alleles, and that's called a knock-in. In one form of knockout experiment involving yeast, for instance, the insertion or replacement of a gene on the chromosome is used to create recombinant strains for genetic analysis. Now, within these recombinant strains, replacing the target gene with a selectable marker is the most useful thing that can be obtained. And that produces a loss of function allele because you replaced the gene with something else. And it also gives you the ability to track the products of that promoter, for instance, that can then be followed in this particular knockout strain of yeast. Should the above mechanisms not be conducive to your organism of study, then there are some alternatives available sometimes. In some cases, when you can't create these loss of function mutations using knockouts, there are some alternative mechanisms that can be used. Once again, transposable elements or parts of transposable elements can be utilized to mutate any gene in any organism. And those collections then can be screened for insertion mutations within the desired gene or genes. Once you have your gene knockout libraries, then the method of screening them to find the gene of interest is pretty standard these days. Using PCR-based techniques, the libraries can be screened. What you require is a primer specific to the gene that you're interested in finding, and one specific to the inserted DNA, whether it be a transposon or exogenous piece of DNA or part of a plant plasmid. It doesn't matter. The method is very similar. So in organisms such as Drosophila and Arabidopsis, which is the weed representing a plant, many strains have now been classified and they are available, and in each strain we know what the genetic arrangement of the gene and its allele as well as the insert is, and these can simply be ordered from a catalog that's available. The technique works as illustrated in figure 14.11. We have different strains of yeast lines or plant lines available to us. In fact, many hundreds and thousands, in this case 100,000, and each one has had a different chromosome that has on average incorporated a random piece of transposable element in this example. Once you receive your library, you generate your primers. In this case, they're indicated here as G1 and G2. And then you also generate a second set of primers that are designed at T1 and T2. These primers are then used in different wells of a PCR apparatus, and simultaneously you are able to use PCR to determine the likelihood of your fragment of interest lying inside your gene. So the key here is the G primers, the G1 and the G2, which are specific to your experiment. The T1 and T2 would be general primers available to amplify all 100,000 clones if they are in an appropriate configuration. So imagine that in the majority of these cases, the insert has gone into another gene, not the gene that you're interested in, or into another location within the genome of the yeast. In those cases, the transposable element will be somewhere else. And for your gene, target gene, you'll only have this configuration. So when this is subject to PCR, you get a fragment corresponding to the distance between G1 and G2, and that's indicated right here. So for the vast majority of your tests, you would expect to see this result. There will be nothing produced from the other combination of primers. If you're lucky, somewhere in the library, and there should be a place where the primers are aligned in an appropriate configuration because the transposable element has gone in this particular example at this location in this intron 2. If that's the case, then the PCR will produce three types of fragments. They'll produce the original fragment between the outside primers, but that fragment will be longer than in the non-transposed gene, and that's indicated by having a fragment that's bigger. So this indicates the gene carrying a transposon somewhere along its length. And then, depending on the position at which the transposon did go in, you will have between T1 and G1, a fragment, and another fragment between T2 and G2. And this indicates the location of the insert within the gene and the fact that the gene does have an insert. Therefore, whichever well in your PCR gave you this result, that would be the clone that you amplify and use in your research, because that contains a insertional mutation into your gene. Moving on to using other known techniques to find and inactivate genes, one natural system relies on something known as RNAi, or RNA interference. RNA interference has become a tool to silence any gene of interest simply by making a double-stranded RNA molecule into a hairpin type of shape. And once you make something into a hairpin, it then becomes fodder for a protective mechanism that operates within cells, either to fight off viruses or to naturally silence repetitive DNA or genes that are not needed in high dosage. This system is best understood with the aid of a figure, and 14.12 allows us to see that. So let's start at the beginning and assume that double-stranded RNA has been injected into this plant cell by some mechanism of introduction. Once that double-stranded RNA has been identified by DICER, DICER will cut up that piece of double-stranded RNA on both sides by trimming it down to about a size of between 21 and 24 base pairs long. Then one of the strands will be digested away completely. So the only thing that dicer holds onto, dicer is a protein, the only thing dicer holds onto is a small fragment of single-stranded RNA. Then the Argonaut protein associates with the RNA single-stranded fragment, and then it goes hunting. It goes hunting for anything that matches in the cytosol, anything that matches in terms of messenger RNA, the internal component that it carries. And once it finds a suitable match, then the messenger RNA is cleaved and degraded. So this is a way of silencing messenger RNA before it gets to the ribosome to be encoded into protein, an intercept mission. You can see we have 1A, 1B, and 1C in this figure because the pathway can be joined from 3 different directions. But once it's joined, the outcome is the same. 1C is particularly interesting in that this RNA is a hairpin. A double-strand hairpin is formed by transcribing genes. So this is a natural intercept mission that will intercept its own messenger RNA that's made by a different fragment of DNA. So it's a regulatory system in this case. In this case, in 1B, it will be part of an immune system to prevent the rapid growth of infected viral RNAs. And then number 3, we can use it as a technology. Employing this system, large-scale reverse genetic screens may be performed. An alternative methodology that does not allow those particular organisms to be subject to transformation by external DNA sources. We can utilize a method called TILling. TILling simply stands for Targeted Induced Local Lesions in Genomes. It's just an acronym for something that some scientists did. And basically what you're doing in this case is you're introducing transposable elements or parts of transposable elements or other forms of genetic markers are introduced into the organism in a random fashion. So by using, in this example, transposable elements of plants or parts of transposable elements, tDNA, you're able to generate many strains of mutant. Each strain is normally going to be present in the heterozygous condition. So all you do is breed them together, self-fertilize, and you look at the next generation. And in the next generation, known as the M2, we're looking at Mendelian Punnett square. So we have 4 boxes, and those 4 boxes represent 1, the homozygous recessive mutation, and the other 3 would be the homozygous and the heterozygous conditions for the dominant wild type. And that's indicated for example 3 here with these small labels, just for example 3. The others would be similar, but their positioning would change. So this would be the homozygous dominant wild type. These 2 would be the heterozygotes that contain 1 copy of the mutant allele in the recessive configuration. And this one here would be the homozygous recessive double mutant. And this is the one that would then be utilized for further research. Once the breeding experiments have identified the source of your gene mutation, the next step is more technological. In this case, we design PCR primers for the gene that we're interested in, the target gene. And depending on the nature of DNA hybridization, this is important now. If DNA double helix is separated by heat and then allowed to come back together again, it will form a double helix based on sequence. If we have a heterozygous condition inside the mutant, then the 2 fragments may come together, but at the location of the mutation, and this is a point mutation for instance, they will generate a hybrid molecule between the wild type chromosome and the mutant chromosome. And because they've generated this heteroduplex, the heteroduplex can be targeted by enzymes that cleave areas of small mismatch within DNA. So cell 1 is an endonuclease that travels along DNA looking for any small bubbles because of mismatched base pairs. And then it cuts the DNA right there. So the result is, if your particular strain of mutant happens to have just wild type DNA at the target gene, then all it will produce is homo duplexes. And there will be no heteroduplex. That will be identified by these PCR primers as a single band. If, however, your particular strain that you happen to think contains the insert, that will generate heteroduplexes, and they will be cut in addition to generating homoduplexes. And when you cut those with the cell 1, you'll get multiple bands. And this is a telltale sign that strain number 3 contains the desired target gene with an insert. All these are mechanisms that are utilized by people working in reverse genetics. So the idea is to isolate your gene first, and then from the gene deduce its amino acid sequence, and then utilizing computer programs or other types of genetic manipulation to find out what mutant effect they would have on the host. Moving on to the last part of the chapter, where we're using technology exclusively to generate transgenes. So transgenes, as we said before, are hybrids made by scientists, and their goal is to construct these DNA molecules so that information can be revealed about the workings of complex systems. In most cases, the regulatory sequence of one gene is tethered to the coding sequence of another gene. So you make something which is called a chimera. A chimera is a hybrid between two animals. In this case, the animal here will be the regulatory sequence, and the second animal would be the coding sequence of a different gene. Or you can take two coding sequences from two different genes and tie them together to see if there's a gain of function. If somehow you can include an external gene product that can be followed during the experiment, then you are using reporter genes. The reporter gene simply shows itself in some form of chemical change, like a color change, as we'll see shortly, or the production of an enzyme. That can then give you the location and timing of when that particular gene combination has become activated. And that's very useful for narrowing down what's happening. You can also generate gain of function alleles that are used to gain further information about the consequences of misexpression. With respect to reporter genes, they come in two flavors. One are transcriptional fusion genes. All you're doing there is you're taking the regulatory sequence of a gene of interest and fusing it to the reporter gene. And that will tell you whenever that gene is supposed to have been turned on and off, because you're monitoring the switch. The switch is the regulatory portion. And due to the pattern of the reporter gene, that will tell you when that switch is on and by how much. Alternatively, you may go for a translational fusion, where the regulatory and coding sequences are fused together so that they do become functional and produce a product that can then be visualized within the cell. We'll see examples of this very shortly. In the top panel, we have a typical arrangement of a small gene along a eukaryotic chromosome. And you should be able to identify each of these parts from your previous understanding. In the case of a transcriptional fusion, we've taken the gene out and replaced it with our reporter gene, which has all the components necessary to use the same regulatory sequence as the gene of interest, but this time, instead of producing a product of this gene, it produces a product of our reporter gene. That product can then be traced within the tissue. In a translational fusion, we go one step further. We keep our original gene and we fuse to the end of that our reporter gene. And we keep the construct so that it can be translated into a hybrid polypeptide. Hopefully, the front end will still work with the back end independently, so the front end can do its normal job inside the cell. The back end can report the location of this protein sometimes tweaking is necessary in order to get the two components to work in stereochemistry. Two systems have become paramount when utilizing reporter genes. One has to do with xGAL and the production of beta-galactosidase. You may be familiar with this from bacterial lacoprons. Our discovery of the bacterial lacopron has led to this particular branch of reporter gene investigation. Anyhow, what happens is that the LAC-Z gene is used as a reporter gene, and it produces, when it's transcribed and translated, it produces in cells beta-galactosidase. That enzyme can break down not just normal lactose, but also other molecules that then lead to different colored products. So if you feed this molecule to this enzyme, it will produce a product from ONPG that is yellow. If, however, you feed this enzyme xGAL, it will produce a blue product, and respectively in that location within that tissue. And we can see examples of that in these panels. So here we have a worm, C. elegans, and the LAC-Z reporter gene is revealing its activity in these cells right here in the anterior part of the worm. In the case of plants, we have the same blue color generated here, indicating that in this tissue, in this plant, the reporter gene is active. Unfortunately, we can't use xGAL in plants because they make their own beta-galactosidase. So in response, scientists developed an alternative system that can be used in plants, which uses the conversion of xGLUC into a blue product in the presence of an alternative enzyme called beta-glucuronidase. An alternative strategy is to use bioluminescence. Bioluminescence simply uses light that's shone on the tissue, and in return, the tissue lights up in various places that contain the reporter gene. So luciferase is the enzyme that is made naturally in fireflies that allows the fireflies to emit light in pulses. And the chemical reaction that takes place in those specialized cells in the abdomen of the firefly is where luciferin and ATP are combined, and they generate this light-emitting chemical reaction. A similar reaction takes place in the ocean in bioluminescence there. Many creatures can generate bioluminescence in the deep ocean, but the jellyfish has taken this to a suitable level in its life cycle. It uses green fluorescent protein, GFP. Under natural environmental conditions, ultraviolet radiation hitting the tissue of the jellyfish results in emission of wavelengths of light that can be detected by human eyesight. In order to overcome the dangers of using ultraviolet light, scientists have modified the natural green fluorescent protein to now respond to lower energies of light, such as blue light. Interestingly, more recently, variant alleles have been engineered that now emit various colors of fluorescence, not just green fluorescence. So this is the typical bioluminescence generated by green fluorescent protein. And in this mouse, just the eyes contain the fluorescent green protein. And when you shine normal light on this mouse, you can see the eyes light up. And these are the photoreceptors in the back of the eye. They contain the actual reporter gene. And in further experiments of this nature, you can actually utilize different versions, different alleles of the reporter gene to generate different wavelengths of bioluminescence. So once again, this is a more magnified view of another experiment in which the retina of the mouse is expressing different types of fluorescent green protein. Not just the effects of mutant alleles can be investigated within genes, we can also use these reporter genes to investigate the regulatory sequences themselves. Fragments of the regulatory region are attached to the reporter gene, and then by generating different types of mutations along the regulatory region, i.e. by deletion analysis, we can pinpoint the region which is important in controlling the activity of that gene. In the case of this particular example involving the Eve gene, even skipped, we're looking at not the gene itself, but the promoter of that gene. And we know that the promoter region of that chromosome can be digested with different enzymes. And by using those enzymes in various combinations, we're able to make these various constructs. So the very top one is the wild type. And of course, that works in expressing under normal conditions in the normal tissues, in these stripes. And then different mutations may or may not work in different stripes. These results reveal that this patch of DNA upstream of the transcriptional start site is very important, and this patch of DNA is also important. But under different circumstances, one patch is used for stripe number 3, and another patch of the promoter is used for stripe number 2. So if you move this around, then you have a loss of function of the reporter gene. If you mutate this region, you have a loss of function of the reporter gene. And if you delete this region, then you have a loss of function of the reporter gene. So that suggests that this region of the promoter must be binding something that helps transcription of the normal gene. The final part of the chapter talks about enhancer trapping, yet another technique that's come to the front in research. In enhancer trapping, the goal is to understand the role of a regulatory region, maybe an enhancer or a silencer, in its expression of a particular gene. In a simple scenario, we have, for instance, this part of the chromosome that has a regulatory region in yellow, and that regulatory region folds the DNA back onto the promoter and expresses this particular gene. And that gene produces a protein that has a normal outcome on the cell. In enhancer trapping, what we tend to do is to manufacture this piece of DNA in the laboratory, and then we incubate this with this system here. And the goal is to get these fragments randomly inserted throughout the genome of the organism of interest. In some cases, because of random insertion, we may end up with our reporter construct, the enhancer trap, going in here between the gene and its enhancer. And the goal here is to produce something from the reporter gene. And if the enhancer is working normally, it will then fold the DNA onto this promoter rather than the original promoter that was used at the beginning of the endogenous gene. In this way, we have concluded that this region of the chromosome is very important in regulating something close by. Therefore, this must be an enhancer. If we see a downregulation in the activity of this gene, the reporter gene, then we can say that this is a silencer. So this is a rudimentary way of discovering the location of regulatory DNA. The outcomes of our research on genes and regulatory regions has led us to start producing chimeric gene fusions, as we mentioned earlier, where we can take any regulatory region of a gene and fuse that to the coding part of the gene. An example of that that's been used in many genetics textbooks is the ability to grow eyes on different parts of the body of a fly. Understanding the biology of the fly is the primary goal of this research, but it shows that master genes can be activated en masse by putting them into places within the body of a fly where they're not supposed to be present. In this case, the Alleles gene has been expressed in antenna and imaginal discs of the leg, even though those eyes don't functionally link back to the brain because there's no neural circuits. They are present in full anatomy.